So, principal sir, shall we start, sir?
Oh, you can start, madam.
Okay, sir. Good morning, respected principal sir. Our distinguished speaker of the day, Dr. G. Ramana Reddy, sir, professor and head, Department of Pharmaceutics and Biotechnology, KVSR Siddhartha College of Pharmaceutical Sciences, Vijayawada, Andhra Pradesh, India, and dear all the participants, a very warm good morning. And this is Harika, Assistant Professor in Department of Pharmaceutics and convener of this faculty development program. It gives me an immense pleasure to welcome you all to the day 3 of the 1 week international online FDP program on cutting edges perspectives in pharmacy, translating molecules into the medicines. This FDP serves as an excellent platform to explore new scientific perspectives and bridge the gap between molecular research and pharmaceutical application. Today, we are honored to have Dr. G. Ramana Reddy sir, who will deliver a lecture on colon targeted delivery of 5-fluorouracil using ethyl cellulose coated pectin alginate microspheres, formulations, and in vitro characterization. His extensive research expertise in drug delivery systems will certainly enrich our understandings of novel formulations and strategies. Now, I kindly request our principal, sir, to deliver the introductory address for today's section. Sir, please.
Eminent research person of today's session, Dr. G. Ramana Garu, uh, Professor and Head, Department of Pharmaceutics, KVS Siddhartha College of Pharmaceutical Sciences, Vijayawada, Andhra Pradesh. Respected faculty participants from various pharmacy colleges, a very pleasant morning to you all. It gives me immense pleasure to welcome you to the third day of the 1 week international online faculty development program on cutting edge perspectives in pharmacy, translational molecules into medicines, organized by VV Institute of Pharmaceutical Sciences, Gudlavalledu, in association with the Association of Pharmaceutical Teachers of India, APTI Central Bangalore, from 13th to 18th October 2025. Today we are privileged to with us Dr. G. Ramana Reddy Garu. He is a very good academician and senior most professor in the field of pharmacy, and Sadi is also having a vast experience in the teaching profession, and also he is a very good researcher also. And today Sadi will be delivering an enlightful lecture on colon targeted delivery of 5-fluorouracil using ethyl cellulose coated pectin alginate microspheres formulation and in vitro characterization. The topic mainly focuses on developing an efficient drug delivery system aimed at site-specific release of 5-fluorouracil in the colon for effective colorectal cancer therapy. The study employs natural polysaccharides such as pectin and alginate to form biodegradable microspheres, which are further coated with ethyl cellulose to ensure controlled and delayed drug release. Through systematic formulation and in vitro characterization, including particle size analysis, entrapment efficiency, and drug release profiling, the study demonstrates the potential of this approach to minimize systemic toxicity and enhance therapeutic efficacy. This lecture highlights an innovative strategy in polymer-based drug delivery for improving the clinical performance of anti-cancer drugs. On behalf of BV Institute of Pharmaceutical Sciences, Gudlavalleru, and APTI Central Bangalore, I extend our sincere gratitude to Dr. G. Ramanagaru for graciously sharing his valuable time and expertise for academic enrichment of our faculty members. Thank you all for your enthusiastic participation. Now I invite Ms. M. Harika Garu, convener of this FDP, to proceed with today's program. Over to Harika Garu.
Thank you, sir. Next, I would like to invite Ms. T. Kundana Madam, Assistant Professor in VVIPS, to formally introduce today's distinguished speaker, Dr. G. Ramana Reddy Garu. Madam.
Good morning, one and all. This is Kundana, working as Assistant Professor in the Department of Pharmaceutical Analysis. It's my immense pleasure to introduce today's speaker, Dr. G. Ramana, Professor and Head Department of Pharmaceutics and Biotechnology. He is a distinguished academician and researcher with over 28 years of teaching and research experience in pharmaceutical sciences. He holds a PhD in pharmaceutical analysis from Acharya Nagarjuna University and an MPharm in pharmaceutics. His expertise includes novel drug delivery systems, solubility enhancement, and pharmacological evaluation of herbal formulations. He has guided over 50 postgraduate and research scholars and supervised multiple PhD projects. Sir has published numerous papers in reputed national and international journals and serves as an editorial board member and reviewer for recognized scientific journals. He has contributed to scientific innovation with several patents and a UK intellectual property design patent to his credit. He is the author of 3 textbooks and a recipient of multiple prestigious awards, including the Bharat Shiksha Ratan Award 2016, Vaidya Shiromani Award 2016, Best Achiever Award 2022, and Pride of India Award 2025. A dedicated academic leader, Sir continues to inspire young researchers and students, significantly contributing to education and healthcare sector. Thank you all.
Yeah, thank you, Kundana, for your nice introduction. So first of all, I would like to thank.
Thank you, Kundana madam. Now I request Dr. G. Ramana Reddy sir to take over the section and enlighten the participants with his valuable presentation. Sir, please.
Okay, I will share the screen.
Yes, sir.
Is it visible?
Yes, sir.
Madam, is it moving?
Yes, sir. Yes, sir.
Okay.
Okay, sir.
So first of all, I would like to thank my friend, the principal of VV Institute of Pharmaceutical Sciences, Dr. Lakshman Rao Garu, and the management of VV Institute of Pharmaceutical Sciences, and Dr. Ramina, HOD, Department of Pharmacology, and Dr. Harika, coordinator of this FDP program, and Srimati Kundana for nice introduction for me. Then other faculty members. So, and dear FDP participants, very, very good morning to all of you. So actually, the main aim of this FDP is a professional development initiative designed for faculty members, focusing on enhancing their teaching, research, and leadership skills. So this program definitely, these often causes areas like pedagogical techniques, research methodologies, and soft skills. So definitely this type of FDP is very useful to the young faculty minds. So please utilize these opportunities and acquire knowledge and improve our research and teaching skills. So actually, this program, 1 week international faculty development program on cutting edge perspective in pharmacy, translating molecules into medicines, is a very novel theme. So definitely the other resource persons will fulfill this title. So before entering into this one, so I am working here since from 24 years in the KVS at Siddhartha College of Pharmaceutical Sciences, Vijayawada. This is the first private pharmacy college in combined Andhra Pradesh. It was established in 1993-94. We are offering B Pharmacy 100 seats and Pharmacy program, and M Pharmacy 3 branches: pharmaceutics and pharmaceutical analysis and regulatory affairs. So especially in Vijayawada means, so all of you know the goddess Kanakadurga Devi will give blessings to all the members who is staying in Vijayawada and surrounding. So if you have any problem, anything, if you worship the goddess Kanakadurga Devi, definitely she will fulfill all these dreams and your desires. So when you comes to Vijayawada, so definitely you visit Kanakadurga Devi and take blessings from her. Then coming to today's talk. So it is my research work we done at our college. Based on this work, I got some patent also and published in good indexed journals also. That is colon targeted delivery of 5-fluorouracil using ethyl cellulose coated pectin alginate microspheres, the formulation and characterization aspects. So first, before entering into this one, what is the necessity of delivering the drugs to the colon? So actually the colon is a part of our gastrointestinal tract. So if you take in conventional drug delivery systems or the controlled drug delivery system, if you have taken the formulation through oral route, after entering through the oral cavity, it enters into the stomach. In case of conventional drug delivery system, there it undergoes rapid disintegration and rapid dissolution, then it undergoes absorption. So here the rate of absorption is the rate limiting step in case of conventional drug delivery systems. So the main drawback of this conventional drug delivery systems is because of this rapid absorption, so it goes above the toxic range in the plasma. So that's why it produces major adverse reactions. So to overcome this problem, so controlled drug delivery systems have been developed. So the controlled drug delivery systems, the rate limiting step is the rate of drug release from the device. So that's why you can minimize the adverse reactions. It's never cross the toxic range in the plasma. So that's why you can minimize the adverse reactions. But in case of conventional as well as in the controlled drug delivery systems, so the major drawback is it reaches the target site as well as it reaches the non-target site also. Because of the reaching of the drug molecules into the non-target site, it produces many adverse reactions. I will give one simple example. If you take salbutamol sulfate you have taken by oral route in the form of tablets, so it requires 5 mg. So that means 5,000 micrograms is required to elicit the therapeutic effect. So once it enters into the target site, especially the beta-2 receptors in the bronchial smooth muscles, it also reaches the non-target site. Because of reaching of the drug molecule salbutamol sulfate into the non-target site, it produces shivering, especially of the hands. At the same drug, if you have taken by pulmonary route, just 50 micrograms or 100 micrograms is enough to elicit the therapeutic effect. So directly it enters into the target site, that is the beta-2 receptors of the bronchial smooth muscles. So you can minimize the adverse reaction. There is no tremors of the hands when you have taken by pulmonary route. So that's why in case of cancer, if you are given all the anti-cancer drugs are the cytotoxic drugs. So if you are given these cytotoxic drugs by either controlled route or by a conventional route, definitely it reaches the this healthy tissue also. It damages the healthy tissue. So that's why you have to go for the targeted drug delivery system. So these targeted drug delivery systems, in this case, the delivery system especially it reaches the only the target site. It may not goes to the non-target site. So based on this concept, so we have developed this colon targeted drug delivery system by utilizing these microspheres. So these microspheres I have opted a simple method, that is the external ionic gelation technique. In this design, the drug should not release in the upper GIT tract. Either in the stomach should not dissolve or should not release the drug. Then in the small intestine also in minimum quantity can release. So once it reaches the colon, so there the different atmosphere is present when compared to stomach and the small intestine. The pH is different and different enzyme system is present and different microflora is present. So based on this, so this device will be triggered and releases the drug. So here I have selected the 5-fluorouracil. This is a commonly used for the colorectal cases. And ethyl cellulose, it will prevent the drug release in the upper gastrointestinal tract. Then this pectin alginate, the pectin, especially the pectinase enzyme, in the presence of pectinase enzyme only, it allows to release the drug. So the pectinase enzyme is present in the colon region only. So once it passes the stomach, once it pushes to the small intestine, there is no drug release. Then it pushes to the colon, where in the presence of pectinase enzyme, so the pectin is going to degrade, then it releases the drug. We can see the maximum abrupt release of the drug can observe in the colon, and it can treat the local diseases of the colon like ulcerative colitis and also amoebiasis and also colorectal cancer and also Crohn's disease. So these kind of irritable bowel disease or the syndrome, this kind of local disorders, local diseases which are present in the colon can be treated very effectively. So this is the simple rationale behind the selection of this work. See here, the colon targeted drug delivery system means the drug should not release either in the stomach or in the small intestine, then it should enters into the colon. So that's why these are called the colon targeted drug delivery system. So when compared to small intestine and this one stomach, so this is the large organ. So you have different parts of the large intestine. So this is the ascending colon, transverse colon, and descending colon, sigmoid colon. Finally, this is the rectum. Okay, so this is having many many enzymes, different enzymes is present. So many bacteria flora is present. So here the residence time is also more when compared to stomach and the small intestine. So some drugs can better absorb in presence of these conditions. So that's why here see these high core containing the drug formulation. So this is covered with entry coat. So when it enters into the stomach, so because of the presence of entry coating, the device will not start to release the drug. Once it enters into the small intestine, here the alkaline pH around 6.8 pH. So the entry coating slowly will be dissolved. Only minimum amount of the drug may be possible. So here the core containing the drug will push it to the large intestine. So in the presence of the enzymes or bacteria, or because of these conditions, here the polymer will be triggered and the drug will be released. This is the simple concept of drug delivery strategies to the colon areas. So why should target the colon? So to treat the local diseases and to treat the systemic sensitivity drugs, and also for the getting the chronomodulated release, the chronomodulated or time release approach. See here, in case of local diseases, mainly the commonly nowadays, if you observe in the some hospitals, especially the gastroenterologist division, even some children's 10 years, 15 years, they are suffering with many complications like gastric ulcers. And also recently, many cases we are observing that is the Crohn's disease. Crohn's disease. So why? Because of our lifestyle only, these diseases are going to get in the children's in the lower age only. So this Crohn's disease mainly is the inflammation of the large intestine. See here, you can observe many of these things. So there is a thickened wall. There is an abdominal pain will be there. So frequent bowel evacuation also will be there. So if you are not treated initially, it may lead to cancer also, especially in outside food. Those that are taking the outside food, so they are using the tasting salt. Especially one reason is the tasting salt is the main reason for causing the Crohn's disease, and also taking up this one unhealthy food like chips and uncle chips. This is a packed food. This also causes the Crohn's disease, and also this is the ulcerative colitis. That is the ulcers in the large intestine. This is also one more reason. So irritable bowel syndrome. Irritable bowel syndrome. So in this case, the abnormal contractions of the this large intestine. So in this is the sometimes there is the frequency of defecation will be more. So repeatedly it will come in small quantity. So this is also one reason. That is abdominal pain, diarrhea, and in some cases constipation and muscle cramping, abdominal muscle cramping, mucus in the fecus, and also the constipation, bloating, and gas. These are some of the symptoms of irritable bowel syndrome. So these also can be treated with the help of colon targeting. And one more thing is the colorectal cancer. The colorectal cancer nowadays, becomes more common, especially in the long back in the Western countries, more because they will take the raw meat, everything. So in India also nowadays, these cases are going to increase. So especially this malignant tumor, that is this is uncontrolled cell growth in the area of this is the descending colon or somewhere. So that may be called as a colorectal cancer. To treat this colorectal cancer, so some anti-cancer drugs can be administered, can be in the form of a colon targeted drug delivery systems. So today's my attempt that what is the I am going to deliver. So that is the 5-fluorouracil. So definitely that is effectively enters into the large intestine colon, and it can treat the malignant tumor which is located in the large intestine. And also some systemic delivery of sensitive drugs also can be delivered. So those drugs like proteins and peptides, so this degrade in the SDK region or the small intestine enzymatically. So these kind of drugs also can be delivered into the large intestine. Their presence of different enzymes, these peptides and proteins can be absorbed into the systemic circulation. And also chronomodulated drug delivery systems also can be formulated. So mainly some diseases will aggravate, will worse at certain times. For example, if you take asthma in the early hours, it will produce a severe effect. Heart attacks in the early hours, inflammation, especially the arthritis. Arthritis is also in the early morning. So these type of these things is called the chronomodulated drug delivery systems can be formulated. So after some lag time, it will start to release the drug. So can be effectively treated or the control the so these type of diseases changes with the rhythms. This is also possible with the colon targeted drug delivery systems. So what are the approaches for colon targeting? Is various approaches are there. So I have used the one approach, that is I have used the this one pectin polymer. The pectin, in the presence of pectinase enzyme, that enzyme is present in the colon only. In the presence of that one, so this pectin will be stimulated and it will start to release the drug. Like that, different approaches are there. One approach is the prodrug or chemical conjugate. In this case, once it reaches the large intestine, because of the bacterial enzymes, this cross bridge will be cleaved and releases the drug. And another approach is also time-dependent systems like a pulsing cap and chronotropic systems. So after some delay of release, the drug will be released specifically after some lag time in the colon. Microbial triggered systems. So this is the presence of microorganism bacteria only. Some drugs are going to activated in the colon. Enzyme sensitive systems. This is our thing. Use of polysaccharides degraded by the colonic enzymes. The polymers like pectin, chitosan, guar gum, and dextran commonly used in the presence of the enzyme system in the colon. So it will be digested and it will be degraded and it will be triggered to release the drug. Okay, that are the approaches. So what are the suitable drugs? What kind of drugs are suitable for colon targeting? So initially I have discussed. So for system, for local action and also for systemic action also can be used. So most widely these colon targeting is useful for the local action of some diseases which are present in the large intestine. So for example, in the treatment of ulcerative colitis and Crohn's disease, the sulfasalazine and belsalazine and valsalagine. So these type of drugs can be used. Mesalazine for the treatment of irritable or the inflammatory bowel disease, and the budesonide. It also can be used for this ulcerative colitis. The colon targeting approach is the pH dependent release, and also prednisolone can be used in the treatment of inflammatory bowel disease. Metronidazole for the amoebiasis, naproxen for the colon cancer chemo prevention, diclofenac sodium 5-fluorouracil, that is the colorectal cancer, and the biodegradable polymer based system. Cisplatin, and this is for carcinoma of the colon, and capecitabine, metastatic colorectal cancer, and ibuprofen, dexamethasone. Finally, the curcumin also can be delivered into the colon. It is effectively controls the irritable bowel syndrome and the colon cancer. So what is the object of the present study? So now I will enter into my research, this one. The main object of the present investigation is to prepare and evaluate the colon-specific ethyl cellulose coated pectin alginate microspheres of 5-fluorouracil for the treatment of colon cancer. See, before entering into this one, just I will briefly explain how the cancer is going to develop, what is the final stage, what is metastasis, this one. So initially there is no symptoms for a malignant tumor until it reaches the stage 3 or stage 4. In some cases, especially in breast cancer, by physically examining the breast, you can find any marks. But in other cases, more or less there is no symptoms up to once it reaches the stage 3. Once it enters into the metastasis, so then the symptoms will appear. So in the stage 0, the size of the tumor will be like a dot, like a dot, or you can say a mustard seed size. And once it takes more time to enters into the stage 1, because it gets less amount of the nutrients from the blood circulation. It requires more amount of the iron. So it may take 1 year or some cases 2 years also. Once it enters into the stage 1, now initially it gets blood circulation from the normal blood vessels by passive diffusion. So it gets less amount of the nutrients like oxygen and the iron. So once it reaches the stage 1 or stage 2, it prepares its own blood vessels, like it is called angiogenesis. Once it enters into the angiogenesis, because of vascular endothelial growth factor will be released, so it attracts the blood vessels and now it prepares its own blood vessels. That is called the angiogenesis. Angiogenesis. Harika, is it audible? Hello?
Yes.
Yeah, is it audible?
Yes, sir. Audible, sir.
Yes, sir.
Okay. Shall I continue?
Yes, sir, please.
Okay. So once this tumor enters into the stage 2, the angiogenesis will be developed. Once it enters into the angiogenesis, it gets its own blood circulation, blood vessels, plenty of amount of the oxygen and the iron it will procure. So the rapid growth of the this tumor. So from stage 0 to stage 1, it takes many years. It comes more time. Once
Sir, Dr. Ramana, sir. Unmute yourself, sir. Sir, you have muted, sir. Unmute yourself. Sir, still
Your
it
voice
is
is
in
not
mute only, sir. Just unmute itself. Click on that mic, sir.
okay.
Now it's audible, sir.
ఓకే నేనేమి చేయలేదు, అది ఆటోమేటిక్ గా మైక్ క్లోజ్ అయింది.
Oh, okay, sir. Now it's
షేరింగ్
audible, sir. It is in sharing only, sir. Just you on the PPT down. You have to shift the windows, sir. Down there showing PPT, no?
Okay.
Yes. Yes,
Is
sir.
it
It is
okay?
visible. Okay, sir.
Okay, good.
Okay, sir. Thank you, sir.
So once it enters into the metastasis, so the first site one is the primary tumor. So now these tumor cells will be detached from this tumor and it will be entered into the other areas of the organs. So there it will be large and it develops the one more secondary tumor. So that is process is called the metastasis. Once it enters into the metastasis, it is somewhat difficult to treat also. So our aim is to treat that colorectal cancer by delivering the 5-fluorouracil drug without releasing the drug in the upper GI tract like the stomach and the small intestine. So it has to reach the large intestine. So there only it should release the drug. So this colon targeted drug delivery has the potential to deliver bioactive agents for the treatment of a variety of colonic diseases, including inflammatory bowel disease, can be effectively treated by the local delivery of drugs to the large intestine. So any local diseases can be effectively treated by delivering the drugs into the large intestine. The treatment of colonic diseases such as amoebiasis, Crohn's disease, colitis, and colorectal cancer is particularly improved by their local delivery to the bowel. So to treat effectively these colonic local diseases, a therapeutic concentration of the drug should reach that target. So if the drug is delivered in somewhere, very less amount of the drug may be reached, so ineffective therapeutic action. So if the drug is released, delivery system reaches the colon, then it releases the drug. A huge therapeutic concentration of the drug is available. So effectively it controls the local colon diseases. So by this technique, absorption of the drug from the stomach and small intestine can be minimized until the drug reaches the large intestine. So various drug delivery systems have been designed that delivers the drugs quantitatively to the large bowel and subsequently to trigger the release of the active drug. Because of the presence of that approach, that may be enzymes present in the colon or bacteria or the pH, based by utilizing any one of these mechanisms, the device will be triggered to release the drug. So by utilizing this any one of these mechanism, we can release the drug in the colon. However, for successful colon drug delivery, many physiological barriers must be overcome. The major one being absorption or degradation of the active drug in the upper part of the gastrointestinal tract. This is the major challenge for the formulator to achieve this kind of targeted drug delivery system in the colon. So the drug should not release either in the stomach or in the small intestine. So then only you can say that you can achieve the goal. Among the different approaches to achieve targeted drug delivery releases to the colon, the use of polymers, especially biodegradable by colonic bacteria, holds great promise. So because of this concept only, so we developed that. So especially the polysaccharides in the presence of these bacterial enzymes, these polysaccharides like pectin and this some gums. So these are going to metabolize or the degraded and releases the drug. Based on this approach, various polysaccharides have been investigated for colon-specific drug release. These polysaccharides include pectin, alginate, guargum, amylase, inulin, dextran, and chitosan. So I have selected pectin and alginate. Based on this, we plan to prepare and evaluate the colon-specific ethyl cellulose-coated pectin alginate beads of 5-fluorouracil for the treatment of the colon cancer. The rationale behind for this is ethyl cellulose prevents the drug release in the upper part of the GIT. So here the concept. So first I have prepared the pectin alginate beads. Pectin alginate beads. So this is the first step. The second step, to the core pectin alginate beads by utilizing the phase coagulation technique. It is a part of it is a type of microencapsulation technique. So the ethyl cellulose has been coated. So in the presence of ethyl cellulose, it should not release the drug in the stomach. Definitely, once it reaches the small intestine, in the presence of the alkaline pH, so a minimum amount of the drug may be released. So inside this is the pectin core, pectin alginate core is present. So this pectin core will be activated only in the presence of pectinase enzyme. Where the pectinase enzyme is located, this enzyme is located in the colon region only. There is no pectin enzyme in the small intestine. So a very small minor amount of the drug may be released in the small intestine. So the major amount of the drug is going to be released in the presence of pectinase enzyme, which is present in the colon region. So before entering into the any formulation aspects, the first thing you have to check, you have to determine, you have to check the drug polymer compatibility. So this is a part of pre-formulation studies. So as a part of pre-formulation studies, so you have to check the drug polymer compatibility study. So you can save the time, you can save the money by performing this one. So once without any this pre-formulation compatibility study, once you have developed the formulation, once the evaluation, at the end you found that if you found that there is an incompatibility between the drug and the polymer, so the total time and total money is also waste. So initially, before entering into the any formulation aspects, the initial thing is you have to go for the drug polymer compatibility study. So here you can utilize two techniques by using FTIR spectral study and also differential scanning calorimetric technique, that is the DSC study. So by the help of these two, we can found if any possibility of interaction or not. So in this case, so I have taken FTR spectrum of the pure drug 5-fluorouracil, and also FTR spectrum for the formulation, that is the pectin alginate microspheres. So there is the principal peaks were not deviated. So especially in this case, the spectral revealed that the presence of peaks at 3001 centimeter inverse for NH stretching, and 1716 and 1657 for the C11 stretching, both for the pure drug and in the formulation. So there is no deviation of the changing of the shifting of the principal peaks of the FTR spectrum of pure drug and also in the formulation. So this can clearly suggest that there was no incompatibility between the drug and the polymer. This is the inference of this FTR spectrum. And the DSC study is also very, very important. So here we are getting the endothermic peak. So DSC DSC apparatus we are using. So here we are using the sample 3 mg, and we are increasing the sample thermogram sample at the 10 degrees centigrade per minute in the temperature range of 50 to 350 degrees centigrade. So initial the thermogram of the 5-fluorouracil and thermogram of the formulation, especially that is the F6 formulation. So in this, what is the simple cause is when the pure drug is placed in this DSC apparatus and if it is heated, so because of its melting point, it gives one endothermic peak. So this is a 5-fluorouracil having the melting point around 294. So we got the sharp peak at 294 degrees centigrade. The same thing in the formulation also. This drug has got the endothermic peak at more or less nearer to this one. So there is a peak in 5-fluorouracil at this endothermic peak at this temperature, and in the formulation is also we got the same peak. So it can conclude that so there is no incompatibility between the drug and the polymer. See if this peak in the formulation is disappeared, or if the peak is shifted to 100 degrees centigrade to the other one. So that can says that there is an incompatibility between the drug and the polymer. So this FTR study and the DSC study clearly shows that there is no interaction between the drug and the polymer. So we can go for the further steps. Next one is the important one is the preparation of alginate beads. That is the pectin alginate beads. So here the simple logic. This is the formula we have used. This is the formula. This is the formula. So totally 6 formulations I have developed. So by altering the various parameters. So mainly the concentration of the pectin, concentration of the sodium alginate, and the concentration of the calcium chloride, and the cross-linking time. This is very, very important. All these parameters are very, very important. So the drug concentration is same. The concentration of pectin has been changed to 1 to 6, and the concentration of sodium alginate is same, and the concentration of calcium chloride, and the cross-linking time has been varied. So initially here the simple logic is first you have to dissolve the amount of the drug into the pectin, then go for homogenization, then we'll get some solution. Then this one pectin solution containing the drug is added to the sodium alginate solution. So this solution again mixed thoroughly, then is taken into syringe. Then this one is added into a beaker. This beaker containing the 6% calcium chloride. Here what will happen is the method is called external ionic gelation technique. See, when the sodium alginate enters into the calcium chloride, the calcium divalent calcium ion exchanges with the sodium ion. So now the calcium alginate beads are going to produce. So here the concentration of the polymer is important, and the cross-linking time. The cross-linking time means the time required for the conversion of the calcium displacing the calcium with the sodium. So if the cross-linking time is less, so there is no complete conversion of the sodium into calcium. So if the cross-linking time is more, there is a complete conversion of this cross-linking mechanism is going to happen. Now we'll get very good quality microspheres can be produced. Here, the steering speed is very, very important, and cross-linking time is important, and also the height of this needle to this baker is also very, very important. Commonly you're using the 5 centimeters distance. Then this drop by drop, the drug containing pectin and the sodium alginate is added to the calcium chloride solution with a proper stirring rate. So these are going to calcium alginate beads are going to produce. These calcium alginate beads are very rigid and free flowing. So this is the first step is over. So here the F6 formulation got the good quality calcium alginate beads. So why? Because here the concentration of the pectin is 6%, and also the cross-linking time is also 20 minutes. So the encapsulation efficiency was observed around 69 or 70% for this formulation F6. So this F6 can be considered as a optimized formulation for this one. So in the preparation of this one, the first step is the preparation of core calcium alginate beads by external ionic gelation technique have been developed this calcium alginate beads. And the next step is the coating of the this core pectin calcium alginate beads with ethyl cellulose. So all of you know the ethyl cellulose is resistant to the acidic pH. So that's why when coated this core pectin calcium alginate microspheres with ethyl cellulose, so it will not dissolve in the stomach. Once it enters into the small intestine, the ethyl cellulose slowly will be released. Then the inner core pectin will not release the drug in huge quantities. So once it push it to the small large intestine, there only it is going to release. In the how you are going for the coating of these microspheres with ethyl cellulose is simply use the coacervation phase separation technique. In this case, these microspheres were dispersed in the ethyl acetate containing the ethyl cellulose and span this the surfactant. Then this mixture was agitated with the non-solvent N-hexane. So this ethyl cellulose is going to deposited on the surface of the this pectin microspheres at the core ratio of 1:3. Then this medium was treated for 60 minutes to complete the process of microparticle coating. The coated microspheres were then washed with an excess of hexane, filtered, dried at room temperature, and finally we get the free flowing ethyl cellulose coated alginate pectin alginate microspheres. And next important step, once the microspheres are prepared, so you have to check the encapsulation efficiency or drug entrapment efficiency. So depending upon the procedure what you have utilized during the preparation of these microspheres, so based on that only we will get the percentage drug entrapment efficiency. So in the first cases here, in this first formulation, the encapsulation efficiency is somewhat less, around 50 to 60%. But in case of F6, because of the cross-linking time is more, so because of the stirring speed is also, we have used 1000 RPM for 20 minutes. So we got the entrapment efficiency around more than 70%. So that's why the F6 is our ideal formulation. This is the simple procedure. This is the simple procedure how to determine the drug entrapment efficiency. So the microsphere should be ruptured. So how can you rupture? Means in the presence of phosphate buffer. So stir this one for up to 24 hours. So then after that centrifuge at 1000 RPM, then filter it. The soapponent layer should be collected. So by utilizing the UV analytical method, you can estimate the drug entrapment efficiency. In this case, by utilizing this formula, you can calculate the percentage of the efficiency. So the F6 formula got the higher drug entrapment efficiency. So that was selected for the further studies. And one more thing, in case of this microspheres, anything, so you have to check the surface morphology. Surface morphology. So if the surface of this one is very smooth, so that indicates that the drug is uniformly distributed throughout the polymer matrix. So this is the first image is the 5-fluorouracil microsphere F6, and this is the ethyl cellulose coated microsphere. So finally it shows that the surface is very smooth texture. So what the method you have utilized, that is the corrected one. So that's we got the uniform this. And one more thing, swellability is also very, very important. Swellability is also very, very important. So swellability, what is the importance of this swellability means before the entry coating, without any ethyl cellulose coating, you have to go for the swelling studies. And after ethyl cellulose also, you have to go for the swelling studies. So this clearly shows that the ethyl cellulose coated microspheres also does not show any more degree of more swelling when compared to this one. So this clearly shows that the coating of ethyl cellulose was done properly. So the ethyl cellulose is not degraded, not eroded either in the stomach. And also once it reaches the small intestine only, this is going to dissolve. So the swellability of different microspheres were determined. No significant swelling was observed with ethyl cellulose coated pectin microspheres, whereas compared with the pectin microspheres as shown in the table, thus ensuring better resistance of ethyl cellulose coated microspheres in the upper GI tract to swelling and preventing subsequent drug release at the non-target site. So once it reaches the small intestine, the ethyl cellulose coating will be dissolved. Next, it pushes to the large intestine. There, in the presence of pectinase enzyme, it is going to release. That is the simple concept behind this here. And in vitro drug release study is also very, very important. In vitro drug release studies also have performed. So in the presence of pectinase and in the absence of pectinase also we have performed. So this type 2 dissolution apparatus have been chosen for this one. So in this case, the pH progression medium has been used. Here you have to measure, check that pH progression medium means, so the drug should exposes the acidic pH in the stomach and alkaline pH in the small intestine also, in the different pH, in the large intestine also different pH. So you have to perform these studies, in vitro drug release studies in 3 different pHs. So like 1.2 pH to mimic the stomach pH, and 6.8 to mimic the intestinal pH, and 7.4 pH to mimic the colonal pH. So first 2 hours in the 1.2 pH, and next 2 hours in the intestinal pH, and up to 12 hours in the pH phosphate buffer, that is 7.4 buffer solution in the to mimic the colon atmosphere. So after that, during this dissolution, so collect the samples at different time intervals. Then the 5-fluorouracil release was analyzed using the UV visible spectrophotometer method at the wavelength of 268 nanometers. So this also clearly shows that. So this is the cumulative percentage drug release of 5-fluorouracil from the pectin microspheres in stimulated gastrointestinal fluids, pectin microspheres. So here up to 2 hours on 1.2 pH, up to 4 hours, it may be in the intestinal pH. So once it reaches the colon, because of the presence of the pectin, so more some amount of the drug is may be released. But in case of this one, coated, so here this is the without any this one coating, and this is the with the coating. So initially in the previous case, ethyl cellulose, there is no ethyl cellulose coating. After ethyl cellulose coating, up to 4 hours or 6 hours, there is a little amount of the drug we can absorb the small intestine. Once it reaches the this one large intestine, so huge amount of the drug is going to release. This is the simple concept. In the presence and without the presence of the pectinase enzyme, in the presence of pectinase enzyme, so there is a rapid amount of the drug is going to dissoluted or the released in the colonic region. So then after that, so these dissolution data was fitted into the various in vitro kinetic parameters like zero order and first order and Higuchi model and first model also. So for zero order kinetic model, the time on axis, x-axis, and the cumulative percent drug release on y-axis will get the curve. From that, you can calculate the R square value. And for first order plot, So the time versus log percent drug unreleased will get the slope value that R square value will get, and the Higuchi model. So does it follows the diffusion mechanism or not? So the square root time versus the cumulative percent drug release. So if you get the linear curve that indicates that Higuchi to follows the diffusion mechanism. So to assess what type of mechanism, diffusion mechanism, so you have to go for the Pepper's model based on the n value, what kind of diffusion mechanism it follows. So based on the R square values of this one, so majority of the formulation follows the first order kinetics. So the R square value, especially the F6, is 0.99 R square value. The Higuchi model is 0.9. It means it follows the diffusion mechanism. The n value is 0.74. So when the n value is 0.45 to 0.8, it indicates it follows the anomalous transport mechanism. Anomalous transport mechanism means first it goes for a swelling and followed by the erosion. So this formulation follows this type of erosion mechanism. And finally, you have to go for the stability studies also according to ICH guidelines. The stability studies, so according to ICH guidelines, in the time temperature of 40 to 40 degrees centigrade, relative humidity of 75% for 3 months. So you have to check the physical appearance, size, shape, surface morphology, drug content, in vitro drug release, and DSC studies also. So the stability studies were performed at different temperature and all this one. It showed that there is no significant reduction in the percentage of drug retained in the formulation, and also there was no significant difference in the drug release profile for the sample storage at 40 degrees centigrade and 75% relative humidity. So these are the stable products. And finally, so I can conclude that the challenge, there is a challenge for the formulator is the device, the drug delivery system should not release the drug in the upper GIT tract, like in the stomach and also in the small intestine. So once it reaches the large intestine, there only the drug should be released. So the experimental results demonstrated that ethyl cellulose coated pectin microspheres have the potential to be used as a drug carrier for an effective colon drug delivery system, especially for the 5-fluorouracil to treat the colon or colorectal cancer cases. It is an efficient technique to treat that type of colorectal cancer. Thank you, everyone. If you have any doubts, can clarify. If you want anything, so I will share to your mail IDs also. Please be in touch with me. Thank you. Thank you giving this opportunity to Lakshman Rao and the entire team of VV Institute of Pharmaceutical Sciences, Gudlaveleru. I hope in future also you may conduct this type of programs for the better use of young faculty minds. Thank you.
Yes, sir, definitely. In future also, we will plan to collaborate with you. Thank you,
Yeah,
sir.
sure.
Dear participants, if any queries, you are free to ask first, sir. I think there were no questions. Sir explained such a beautiful way. So, coming to our end of the section, thank you, sir. Thank you, Dr. G. Ramana Reddy, sir, for your highly informative and insightful talk. On behalf of VV Institute of Pharmaceutical Sciences, we express our sincere gratitude and present a token of appreciation in the form of certificates, sir.
Yeah, okay. Thank you.
Thank you, sir. Thank you, sir, for your support and wonderful, highly important, informative talk, sir. Thank you.
Okay, okay. Thank you. God bless you. Bye.
Thank you, sir. Sir, you can leave the section, sir. Thank you, sir. Dear participants, the attendance form will be available in the chat box and please ensure to fill it before leaving the section. Dear participants, the attendance form is available in the chat box. Please ensure to fill it before leaving the section. Thank you all, and we look forward to meeting in your next section, that is tomorrow, day 4. Thank you for your active participation and hope to be continued throughout the FDP program. The attendance is mandatory to get the e-certificate. Thank you everyone. Thank you for your cooperation.